you go, Jared. Tanner, thanks for logging on.
How we doing, Danny?
Doing well so far. Excited for a nice discussion this morning.
Settings here because it's telling me I can't share my screen, or at least turn on my camera. Where's good old Zoom?
Danny, can you hear me?
Yeah, Jared, you're good, Jared.
Okay.
Okay, well, I guess the link went out because we got 6 people on here, so that's good. Look, Tanner, now we got 2 Tanners. I got you twice, Tanner.
I don't know what happened. It kicked me out, and then because I had to switch to allow for the show my camera, so I'm on there twice right now.
Yeah, here, I'll kick the other one out. No, now you're good. All right. Okay, well, I guess we'll get started here in about 6-7 minutes for everybody that's on. So thanks for logging in early. We're expecting a pretty nice crowd today. So hopefully things go smoothly here. If you have any questions, feel free to use the chat. If you want to send things over, we're using a webinar format today. So no one can see you or hear you if you're an attendee. So feel free to continue on your normal business, I guess, and enjoy the show. But if you have questions you want to send in, go for it through the chat or the Q&A options that are on there. On Zoom there. Jared, so what's going on in the valley right now? I drove through a couple days ago and through the south end of the valley, and it looked like almond bloom was still going pretty strong. Is that the same up there in your neighborhood?
It is, yeah. It's up and down the state, a little further down the southern part of the valley, that Arvin, Bakersfield. But yeah, we're in full bloom and petal fall in a lot of places for our almond customers and almond growers. So that's an exciting time. Grapes are right around the corner for give it a couple more weeks. Even some early varieties are starting to push down south. So yeah, we're getting into it. We've been a little dry. We wish we had some more rain, but we're hoping to get a little bit here this next week. But we'll, it's California. Sometimes we get it, sometimes we don't. We just got to learn to deal with it.
Yeah. The I know a lot of times I hear we get those good February rains and things like that, and it slows down applications and people aren't able to get in their fungicides and things like that. I mean, has it been pretty smooth sailing this year with the weather in terms of how bloom's going? People expecting good yields out of it?
The bloom it looks pretty good, I would say, across a lot of the varieties, particularly down south where we're further ahead than, say, Northern California and almond bloom. It does look like dormancy was good. I'd say on the most part, flowering pretty even throughout the tree and the canopy, which is usually a really good sign. And bee weather has been excellent. And almonds need bees to pollinate and be fertilized. So that should be good. We'll know more here in the next, few weeks to a month, to figure out what did stick. There hasn't been a lot of weather, so we haven't had a lot of fungicide go out. I would say less than normal. So not a lot of activity sprayers, and you don't see a lot of helicopter because of rain. You can't get in the field that you'd go by air. So yeah, it's been pretty dry. So yeah, we'll see. It should be pretty good bee weather. So we're hoping yields will be good.
Good. Sounds good. And it's still pretty early for pretty much everything else. Almonds are one of the first things to pop out of dormancy in the valley, right?
Yep. Pistachios are much later. Cherries are starting to go. So we've got cherries in the southern part of the valley that are in bloom. Further north where there's more acreage around that Stockton area, just starting to get going on some early stage 5% popcorn bloom. So we're still early there, but cherries are right behind. And obviously we have early nectarines, some stone fruit that's early. That's plums, things like that are blooming right now for sure.
Very nice. Very nice. It seems like obviously today we're going to talk a lot about stress defense. It seems like we haven't had a whole lot of notable stress, I guess, in the valley up to this point. I mean, it's pretty used to being cold and things like that. You guys had some exciting wind and things, but nothing. I know I've been over there during bloom where it's been dumping 3 inches of rain and
Yeah.
yeah,
Yeah,
and
or
14 degrees.
we get down to 28 degrees or 26 degrees in pockets. Yeah, we haven't had that yet. It always makes you nervous. We're still in a lot of cold weather territory here, early March, even throughout March. So just a couple years ago, we were in full bloom, and there was icicles hanging from flowers and limbs as people would put in water on, trying to keep temperatures warm, as warm as possible. So that's still potential. I don't see anything in the next 2 weeks to get that cold, so we're keeping our fingers crossed that we don't have that.
Yeah. I was talking to Justin the other day, and he sent me a picture of one of his orchards that had a branch of I assume it was almonds that just the blooms were just you couldn't even see the branch in between the flowers. There were so many
Roped.
yeah. Is that so? I mean, and then I've seen others where there's a flower here, a flower there. Is that an indicator of future yield? Is that something that's farmed for? Kind of
Yeah,
what influences that?
sure. You're trying to build carbohydrate stores the best you can so you have more fruitfulness the following year, and repeat that, right? Not have a strong year and have this alternate bearing type situation. So some of that is varietal of where how thick flower can be, and the almond it'll want to hold and retain so much that it feels it can handle and mature. So even if you have a lot of flower on, if you don't have the proper nutrition and water management, keep stress off, you can drop several here in a month and 2 months down the road where you'll lose yield. So you're trying to hold on to that and retain that.
Very nice.
So yeah, and in cherries we call that kind of knuckles. I mean, you'll see that, and the better you go into dormancy, better previous year they had, you'll see that nice consistent flower, roped on. I mean, there's different slang terms that we use, but it gets you excited for sure.
Mm-hmm. Thanks everybody for logging in. We'll get started here in a couple minutes. Gordon, Chris, thank you for joining us here for our panel. So I see you, and looks like we're good there. So we'll give everybody a couple more minutes to get going, to get logged on here. We're still seeing a decent amount of people pouring in, I guess. So we'll get started a couple minutes after the hour. We're using the webinar function today here on Zoom. So if you have any questions, please submit them through the chat or I think there's a question function or something like that. We'll monitor both of those as we can. And yeah, I think that's good. Yeah, we're still getting plenty of people.
What are you guys right in the middle of down in Oxnard? Strawberries going strong? What's the latest?
Strawberries are going strong. Yeah, it's warming up a little bit, so yields are starting to pick up a little bit in strawberries. Starting to see the plants really take off. Kind of getting out of that early season production, more into the higher yields. It's still a little cold. It's been really windy. It's been some problems there. Avocados the market finally came up, so that's been people picking avocados for about going on 2 weeks here, I guess, where the market's been acceptable.
Good.
So that's nice to get the fruit off the tree. It's nice timing. Bloom is starting to go in avocados. Lemons, we're starting to see bud development and push. Vegetables this is full vegetable season in Oxnard, so celery's still going strong. Cabbage, leafy greens, and all the other leafy greens and things like that. So yeah, it's busy time in Oxnard. I was in Salinas this week too, and they're getting going. A lot of plants in the ground, getting their first lettuce crop coming up. Some of them even 6-7 inches across already. So for some of the early lettuce plantings, so it's good. Things are getting moving here on the coast across the board. Of course, Santa Maria's always got something going on. It never stops in Santa Maria. They're that wonderful in between zone. So
Mm-hmm.
yeah, been good. So yeah, it's busy.
That's good. We
Never
like that.
stops in California agriculture.
No, it doesn't.
Surendra, thanks for logging on. I see you here. So
thank you.
So we've got all our panelists and everything. everybody's here. I see a lot of familiar names on this list. Thank you. And thanks for joining us. Jared, you want to get us started?
You bet. Yeah, I want to welcome. My name is Jared Sanner. I'm a Western manager for Redox and live in the Fresno area. I'm happy to be here. I'll be helping, just kind of in the background, helping facilitate. There'll be a lot of people on the call, so just trying to grab questions and make sure we answer those as we move along. And want to thank our guest. Daniel will introduce himself and the others later here. You see the topic there, Redox University. We'll really focus on stress defense and walk you through that. And we're excited to have our participants today. You should have all received a packet, whether digitally or through the mail. In that packet, in that folder, you'll have obviously the schedule and a welcome letter. Our abiotic stress defense guide that we'll be referencing through the discussion. You can reference later. There'll be some solutions as well that you'll find there concerning abiotic stress, and then some multiple redox results of where and how and when, some documentation on some trial work that's been done dealing with stress and some of our technologies. So again, any housekeeping, raise your hand in there. You'll see we're on a webinar format, so you can't just jump in, use those questions, raise your hand. We want to get to those. So participation would be great. There's a couple things there. Thank you, Danny, for throwing that up there. Please use that function. That makes it a great webinar when we do that. If you're on a mobile device, you can, on the app, you can just continue on your day if you're driving and listen through that. It should be pretty seamless. And we encourage you to do that. If you have to get in and out of the truck or away from your computer, you can always do that. And with that, I will, there's our schedule for the day, and we'll get started here. I want to introduce our primary speaker and host who's put this meeting together. Is Dr. Danny Klitich. He received his PhD from UC Davis, where he studied interactions of fertility programs with pest populations. And he definitely has a passion for what he coins, and I've heard him many times, building a stronger plant and doing that through sound input choices like fertility, irrigation, just different inputs across the season, but really with an eye for economic profitability. That would be something that's a driver for him. His passion is working here with Redox as a regional agronomist in the California coast area. And we're really happy to have Danny on our team. He works extensively in avocados, berries, leafy greens, citrus, and wine grapes. I probably left a couple out, Danny, but I think that gives a pretty good description of the various crops here on the California coast that you take care of. So Dr. Danny, take it away.
Thanks, Jared. Really appreciate that intro. So yeah, looking forward to having a wonderful discussion today with everybody. I thought this statement kind of maybe frame the conversation of where I want to take these days. How to grow a profitable crop that's more stress tolerant and resilient without spending all day micromanaging input, slogging through piles of data, or wasting your money, right? So that's everybody's fear is I'm going to try and deal with stress. I got to buy a whole bunch more meters that I'm going to have a bunch of apps on my phone, or I'm going to have to spend a bunch more money on biostimulants and water penetrants and things. And so I wanted to kind of, hopefully throughout the discussion today, we can kind of build a framework of how to think about crop stress in a meaningful way that helps influence how we make our farming decisions. Redox really exists to create passion and excitement around growing plants. And I hope we bring that to you today and give you some fuel, I guess, to help with that, hopefully with your passion and excitement in growing plants. I think that's why a lot of us got into agriculture, is it's fun to grow food and for the world. And I hope that we can help bring you some new concepts and some new frameworks to help build your excitement. So today we're going to start with a panel discussion. We've invited 3 top growers to kind of have a discussion around crop stress. I'm going to like to read you the question to start with here, and then I will introduce our 3 panelists. So crop plants, whether it's strawberries, lettuces, avocados, alfalfa, almonds, whatever, etc., all have genetic potential to achieve a maximum yield. Crop stress, however, limits our ability to grow and produce at that high yield. What tactics and strategies have been used in your farming operations to limit crop stress and drive yields? So that'll be kind of, and I'll read that again after I finish introducing everybody, but I wanted to kind of frame the frame the discussion here around what we're going to talk about. So our first panelist I'd like to introduce is Gordon Kimball. Gordon is a fifth generation farmer in Ventura County, farming over 100 acres of avocados. He is active in the agriculture community as the director of the local groundwater sustainability agency and the current chairman of the board of Limoneira Company. As I'm sure you will see in this discussion, his passion for systems and engineering still shines from his early careers as a mechanical engineer designing Formula One and Indianapolis race cars for teams including McLaren, Ferrari, Benton, Pernelli, Chaparral, and Patrick Racing. So it's my pleasure to introduce Gordon Kimball to the panel. Our next panelist is Chris Height. He has extensive, I can't read today, he has extensive experience in the lab and field analyzing soil samples and making fertility recommendations to support over 10,000 acres of vegetable and other production in the greater Santa Maria area. That's over 32,000 acres of crop production every year. And he does this with Betteravia Farms. If that doesn't sound busy enough, he is also currently pursuing a master's in soil science from Cal Poly San Luis Obispo and likes to run Spartan races on the weekends. So it's my pleasure to thank Chris for joining us today. And our third participant today in our discussion is an expert in all things strawberries with over 1,000 acres of production every year, covering both organic and conventional production systems. There's never a dull moment there. He is a self-described just another farmer, but his business card says he is the Southern Region Strawberry Production Manager for Ryder Brothers, Inc. in the Oxnard area. So it is my pleasure to introduce Tanner Sherman to our discussion. So if you guys wouldn't mind unmuting your microphones, and I'm going to stop sharing my screen so that we all can see kind of the top speakers here. So let me just read this question one more time for everybody. So crop plants, whether it's strawberries, lettuce, avocados, etc., all have genetic potential to achieve a maximum yield. Crop stress, however, limits our ability to grow and produce that high yield. What tactics and strategies have you used in your farming operations to limit crop stress and drive yields? I don't know. Anybody want to offer kind of maybe an example of a stress that they find just that they're dealing with day to day that just keeps coming up? That's not labor. We're talking about stress on the crop, not stress on the farmer. Anybody?
Well, one of the things we deal with is since we have so many acres, we have a lot of different fields that we have to manage irrigation for, and that's going to be one of the biggest things is getting the right amount of water on the right portion of the field at the right time, and trying to get enough water so we don't have to bring all of the pipes back onto that field. So irrigation management and anything that aids in the water retention in the field is going to be one of the big things that helps us out.
Yeah, that's interesting, Chris. I think that water management is almost always key, right? It's our biggest input on any farming operation that's irrigated, and it seems to be it really defines it. It's what am I trying to say? It is the factor that can make or break a crop as if the irrigation is taken care of properly. I know, Gordon, you've had a lot of fun with irrigation lately.
Yeah, and I think globally in avocados, I mean, I look at the vegetable guys and the amount of lab work they do, and presumably it really helps. I think in avocados, most days I feel like we're going avocados kind of like this, and we have the trees don't respond in days or hours unless they're really dry. But in terms of, I think probably our biggest stress that we don't even know or understand is nutrition. It's hard to understand what they really want. There aren't the studies. A lot of the studies on avocados are older in terms of nutrition and what elements, what is the right range for elements. So I think other than the obvious that everybody faces, weather, the heat, the wind, the cold, we have a few stresses in terms of salt buildup, but with sprinklers, at least it's fairly easy to leach that regularly and avoid that. And the biggest fundamental disease is probably Phytophthora cinnamoni. And in that sense, in that area, the new root stocks are much more resistant, and that problem seems to be diminishing. So I think the biggest one in avocados is understanding the right nutrition and getting it to the trees without it getting locked up in the soils.
I didn't pay Gordon to say that, so I really appreciate it. No, I mean, I agree, right? That's the biggest challenge we have in a lot of these systems is making sure that that the plant has what it needs. It's why we grow crops in the ground, is we hope that the ground provides things to the tree, and it does, but then it doesn't always do it when we need it, right? And I think that's one of the things, Chris, in your lab, I mean, that's one of the goals, right, is really seeing what's in the ground.
Yeah, we go out and constantly are checking for nitrate, and then twice a year at least we go out and check all the soils for different nutritional event, what's in there. And before planting, we always go out and try and do another application of whatever needs to be shored up that isn't nitrogen before we go out and try and plant again. So we try and get everything in beforehand. And one of the things that really limits us is the sodium and salt stress too. So trying to see which ones are highest and get that out before we plant, especially in Lompoc.
Yeah, Tanner, what do you? I mean, what's your take on that? With water management and obviously how do you use analytics in your operation to kind of maintain your crop?
Yeah, no, I mean, with us, I think just in general, strawberries have always kind of been overwatered than underwatered, and so for us, it's kind of those irrigations, whether it's through tensiometers, and two is just unfortunately is the quality of water, kind of in the Southern California region, where we typically have high pH water with containing a lot of either calcium or salts as well, sodium. So for the most part, we're always having to have a sulfuric acid machine running with it, and when you're running that, obviously now your soils are getting a lot more sulfur than needed, and so it's just, again, kind of going off of the other two, I mean, just finding that balance between everything. I mean, I know with strawberries, unfortunately, they are a pretty or they're not very tolerable in when it comes to stresses, and they're pretty finicky, and so the littlest things can make or break you on a ranch. And so for us, it's just been that is finding the balance of everything, and trying to leach when you can, but it's kind of hard once you're kind of in the go time of the year. I mean, obviously with strawberries, they're constantly cycling and different fruit set on each plant, so it's not something that you can take a designated time and just run your water and try to leach everything down, because at that point now you've lost a day of fertility, because one plant has flower, green fruit, and red fruit, so it's taking all of its energy and then pushing it to each cycle on that individual plant. And so I'm just trying to find the balance, right? And that's been our biggest thing.
Yeah. What would you, Tanner? What would you say in terms of stresses that are limiting production this year for you guys in strawberries?
Yeah, this year, I mean, just kind of going with the theme of no rain, and it seems like we get a Santa Ana wind event here about every 2 to 3 weeks. So typically when it's one wind event, your plants are going to take a little bit of a beating, and then they kind of recoup and get out of it. And unfortunately for this year, it just kind of seems that because we're getting one every 2 to 3 weeks, it keeps hammering down on the plants, and with that, if you're not maintaining your ranch, keeping your roads wet, you get a lot of dust. With that dust comes two-spot spider mite, which is our biggest predator, I guess you would say, which can easily take over a field. And so, yeah, with these dry conditions, it really flares up the two spot, which, yeah, I mean, and I think it's kind of the theme this year in the Oxnard area that we're all dealing with.
Yeah, and two spot usually comes on much later. Like you're probably about almost a month ahead on brutal two spot pressure, right? It's usually more of a late early
kind of
April.
a April, May deal, and then this year, I mean, we've seen it obviously as early as February, so it's been something we're battling with.
I know you mentioned poor water quality. We have I looked through the list of RCPs we had before this, and we have people from all over the globe, honestly, but a lot of people from outside the kind of the coastal region here. And our water is pretty much is just terrible in terms of a lot of people's standards, right? Our ECs starting out are 1.5 in a lot of cases, 2. I mean, we've got some wells that pushing 3. So yeah. So I think that just for frame of reference for some people that are maybe not familiar with California coastal cropping systems here. So Gordon, what do you think some of your top yield killers this year, or stress events that you guys have been trying to manage around?
Well, the east wind was a yield killer, and the fruit that set last May was on and would be harvested this spring, summer. We had 70-mile-an-hour gusts for over 4 hours, and that I think it put about half the fruit on the ground. So in terms of the tree, it's probably in a way a good thing. It unloaded the tree. We'll see how much of the bud that was going to be flush growth can be converted to bloom growth, and hopefully we'll set a better crop for next year and won't have east wind. But the wind and irrigation, oddly enough, in winters like this with this much east wind, we have a harder time keeping the trees hydrated than we do in the summer when it's 85 and 90 degrees. It's like low humidity, and the wind just sucks the water right out of the ground and right out of the trees. So probably hydration. I mean, the wind you can't control, but the challenge is hydration.
Yeah. Okay. Yeah. It does sounds common in line with kind of some of Tanner's major stressors there as well. So Chris, how about you? What do you see on kind of your guys's wall of villains?
So the two spot, we haven't seen too much of that yet this year, so we're keeping our fingers crossed. But yeah, the sodium is definitely creeping up, and then we're having not as bad as last year, but the diamondback moth has really been putting pressure on us. Been one of the biggest stressors, and we've seen it in green cabbage this year, which it usually isn't, but it mummifies the cauliflower and the broccoli, and especially in the cauliflower, it gets in when the head's forming, and then you don't see it till you get the head apart and you see the worms crawling through it. You gotta toss out that cauliflower, and you just don't know until you open that up. So it's been one of the bigger challenges this year and last year. And those suckers have resistance in just about every single pesticide, and each time you get a wave, you get a different type of resistance. So you never know whether it's going to be good or bad or what. But that's been two of the biggest things.
Yeah. I got a question here from the attendees, I guess, here. So, and if you guys want to participate or send in questions, feel free to shoot them up there in the chat. We'll see if we can get them proposed to our panelists here. So I've got one here that says, in South Africa, our biggest challenge was thrips this year. I'm not sure in what cropping system. I'm gonna go with avocados maybe, but or berries. But yeah, we definitely see a lot of thrips in our systems too. Here, Josh, I know Josh is an avocado grower here. He says wind is their biggest issue. Our comment here from South Africa says strawberries, but generally all crops. So you guys see much thrips issues, Tanner, in strawberries? Or Chris, I guess Chris, you guys have some strawberries too.
We do, yeah. No, I mean, it's something that typically, I mean, yeah, if it's not controlled, or, again, kind of the climate, how it's been with just dry and, yeah, they'll kind of creep up, especially with the dust pickup. And yeah, the biggest thing is that they bronze the fruit. And so with that bronzing, then, I mean, obviously it's not marketable, so we'll end up losing. But knock on wood, so far it hasn't been a huge issue yet. Typically it kind of comes within that April, kind of as things start warming up as well. But no, definitely can be a big issue if not taken care of.
Yeah, yeah. I got a question here in regards to avocados. It says, are most avocado growers still resisting utilizing surface mulching? Gordon, what's your kind of take on the acceptance of mulching within the avocado industry to help with water management and soil health?
I don't know about resisting so much as, I mean, it depends on tree age, but a good, healthy, mature orchard provides its own mulch. And my take on it is it's absolutely critical, because you dig under that mulch, the leaf mulch, and there's a vast system of roots on top of the ground. In fact, we had a pipeline break and washed a significant area of the orchard, and it just washed the loose mulch and the very loose topsoil off. And what was left was just this, I mean, it looked like hair. So I don't know about the acceptance. The part of the problem is, and I've seen it, people use that. Part of the problem is getting the right mulch, because people have used green waste that's been processed, and it turns out very fine, and it actually works like a sponge when it's wet, and it can actually drown the trees, because the avocados actually need to breathe through those roots. So you need a mulch that's open. And I think some people have tried, because processed green waste is getting cheaper and more available, since it can't go to the landfills here in California. But you have to be really careful, that I mean, basically tree trimming chippings are way better than anything else. So I think it's coming. I think more growers are aware of it, but it's not a cure-all. You can't just put organic material on the ground and all of a sudden the trees look better.
Right. Yeah, and I think it does help with stress, right? I mean, it helps with your water holding capacity in the soil. It helps with the weed pressure, robbing your nutrition and water. It helps with soil insulation when it's hot, soil insulation when it's cold. So I think it helps in a lot of ways, but you got to have the right stuff in the right time, in the right place, right? So thank you, anonymous attendee, for sending that. Oh, they're down in Temecula, I guess. So that's great. So I think here's another comment from one of our participants here. Karen says, release beneficials, I assume referring to our spider mite issues. Maybe let's talk about that for a second, because I know I think both you guys utilize beneficials to some extent in your strawberry production systems. And I would say that releasing beneficials is actually a pretty standard practice within berry production to a certain point in the season. Tanner, Chris, one of you guys want to kind of talk about beneficials and how that's kind of used to manage both thrip and mite pressure?
Yeah, no, I mean, the biggest one for us is persimmons mite that goes out. I know some can go as far as Californicus as well. So I mean, there are definitely beneficials that go out. And yeah, no, I mean, and it's something that we're constantly releasing, right? I mean, at some point we also have a budget, and so we do have to go with that. I mean, yeah, obviously the easiest answer would just be throw out as many as you can, but no, we definitely try to control, and that's probably the main source of controlling our spider mite issue is beneficials and through persimmons. It's just, yeah, unfortunately sometimes too is the colonies don't like to establish, and all of a sudden again, just the two spot keep taking over. but it's definitely a standard practice. Yes.
Yeah, that's same here. I've also seen the Californicus dropped out, but usually we use persimmilus mites, and really it's knock the population down with the spray, release the mites to try and keep those up. And then we've also been using the micro sprinklers to try and establish the humidity to try and keep them happier. So
Yeah, because that's a big deal. Is a lot of these beneficial mites, they really they're pretty persnickety on what temperatures they like, and if your climate's not right in the field, they're just a waste of money to some extent, right? Because they're going to go out there and they just won't thrive. You get thriving in a good environment for them. So, but yeah, I mean that's the way the industry is going, because there's not really a whole lot of new acaricides coming onto the market that have great efficacy, and the old ones aren't working as good as they used to, and our solutions are kind of going with what used to be the second string of defense is becoming the primary. That's growing a stronger plant, which we're going to talk about today, and trying to make sure that that we have beneficials present to help us out. So yeah, thanks, Karen. Yeah, so I think we're going to kind of shift here from the panel discussion into kind of the group discussion here a little bit. I'll keep the panelists up here for a second, and maybe for the attendees here, if you could use the chat function and just send in some examples of crop stress that you deal with, maybe that we've talked about now, or that we've had otherwise, or that you've thought of while we're having this discussion. Pull up that chat, just start typing them. I'm just going to kind of, I just kind of want to get, what are we talking about when we talk about crop stress, right? There's a lot of different ones out there, so let's do that now. And then we'll go into our discussion around the physiology of plant stress and kind of what that is. So look at that. There's exactly 0 chat. There's one. Wind followed by heat, and it's a major issue. Thank you. Anybody else? Nobody else has any other. So we had a complete. Derek, here we go. Thank you, Derek. Crop yield is a stress. And sun scorch. So that's an interesting one. Crop yield is a stress. That's I think Gordon alluded to that with losing half his crop onto the ground instead of into bins, and how that was going to improve next year's set because that stress was removed. So that's a good one or interesting one. We got gumminess on nuts results of stress. We got burns on limbs, sunburns. That's a big one on a lot of avocado, and that's true on other crops as well. When we have extreme sun events, we can get burning. Troy, thank you. Salts, heats, so on. John's talking about here on greenhouses. So greenhouse light intensity, right? So we can have when we have a lot of cloudy days, that we can have poor light intensity, and the opposite as well is true. If it's too hot and too much light intensity, we can actually have issues caused there. Jim Wilkinson, we've got heat, water, viruses, disease pressure, absolutely. Sun scald on fruit, cold temperatures at petal fall or small developing fruit size, mealy bugs on citrus. I think that's a lot of these pests, especially the hemipterans, they can just literally suck the energy out of the plant, right? They're not necessarily causing direct damage, but they're literally stressing the plant by pulling out and removing a lot of the sugars and photosynthate production that we have in there. Bryce says, Gordon touched on this some already, but a problem I often see is over irrigating causes I think it's supposed to be anaerobic conditions and induced micronutrient deficiencies. Yep, that's a big one. Water management, coming back to our initial discussion there, was absolutely water management. Stress from Tracy here says stress from red scale following treatment from ACP on citrus. I don't have anybody here that has much citrus, but that's definitely a big one, is that we have these secondary pests popping up. We see that a lot in strawberry production, and where you go out and treat for 2-spotted spider mite and something else starts taking off and causing issues, right? Bagrata bug in broccoli, says Robert. Bob's got mature lemon crops are staying on trees longer. Picking often does not occur until significant drop begins. So I think that's the second one we've had here about the stress of the crop on the tree, right? So a lot of these systems that we're growing in for fruiting crops have successive crops or successive crops on the tree at one time, or at least overlapping phenology of harvests. So avocados, we have fruit on the tree past our flowering date in many cases. That's a big stress. Lemons are constantly fruiting here on the coast. Strawberries are the poster boy for having fruit, flowers, everything going on all the time. And if you have a big fruit set, you're gonna have you have the potential to have a lower fruit set later. So yeah, I've got more over here on Q&A. Sorry. Wind following heat events, loss of flowers from wind damage. That is absolutely happens. That was on citrus they were referring to, but got a bug on broccoli. And then here's maybe Gordon, if you want to take this one. I think this is a question that we deal with a lot in avocados. How do you prepare for the next season when you lose your fruit slash flower crop for this season? Focus on foliage health. What do you think? Kind of what strategy would you go to? And I think maybe we'll take that to the strawberry to Tanner and Chris after, because it's true there too, where we have these big stress events and it maybe not knocks off crop, but we dings us pretty hard. Any thoughts there, Gordon?
I mean, it comes back to, again, having a strong tree and supporting a strong tree. And to me, it's letting the tree decide what it wants to do. If it wants to do foliar flush, or if it wants to build buds for bloom the following season. In terms of the tree, I'm kind of a laissez-faire guy in that I want the tree to decide what it wants to do. I just want to give it all the tools and all the nutrients it needs to do it, and make sure that it's strong enough so that when it makes its instinctive or hereditary choice, whatever it is, it's able to support it. I don't think I'm smart enough to tell the tree what to do yet.
I don't think it would listen in most cases either. I think that's the most frustrating thing about agriculture, right? Is we think we know what we want it to do, but what are you going to do? You start playing with PGRs, it starts getting a little too exciting sometimes. So, yeah, Tanner, Chris, anything you wanted to add to that in terms of kind of managing? I don't know, the crop for the future when we've had a big yield. I know that we deal with this a lot in some strawberry varieties where we have these big gaps, right, in production, where we have a big pick and then it gaps pretty hard, and then we hope it comes back, right, on some of these varieties. So I don't know, do you have anything, strategies or tactics there you've used to kind of deal with that stress? Because this is a stress, right? You have this huge fruit load that pulled all the nutrition out of the plant while it was sizing out, and then you don't have any flowers for that 4-week period or whatever it might be. So any thoughts there?
I think for us, it's mainly, I mean, it kind of comes down on the labor side, but it's getting the, because again, typically after a wind event or whatnot, you'll have some dehydrated fruit, and it's getting that off the vine as soon as possible. I mean, so that the plant does not, continue feeding energy to that fruit that won't be marketable. Same thing, sometimes we'll see some crooked fruit come off of it as well, some cap pacing, and so picking that off as soon as possible, just to, again, at least the plant is sending all of its energy toward the flower or green fruit that will be marketable, and then also the energy to split new crowns and kind of recycle and get going again.
Yeah.
Yeah.
Anything else? Go, Chris.
I was gonna say same thing here, trying to get off the ones that are not marketable and put in the energy into what's actually going to be marketable in the future. And then just making sure you don't have a diseased or weak plant. And as mentioned, not a tree, but making sure you have a strong plant to continue with the growth.
Yeah. Okay. Well, I think that's, yeah, I think we'll cut it at that. I think that's a great discussion. I really appreciate your guys' participation and kind of sharing some of what you do on a day-to-day deal with stress. And I look forward to kind of working with you guys to work on with some of those, and then for the rest of this talk here, where we've kind of helped frame it on kind of what are we talking about for plant stress, and how do we deal with it on a day-to-day. So thank you very much, Tanner, Gordon, Chris. Thank you. Okay. So our next deal here is, I gotta share this screen. Okay, so so the next part of the talk is I'd like to kind of talk a little bit about physiology of plant stress. So I find it helpful to kind of go back to what's going on in the plant so that we can devise a way to help manage that stress, right? So what's the mechanism for that stress? Okay, sorry, I got a chat thing that popped up here on me. So let's go there. So one of the values at Redox is being passionately authentic, creatively driven, and scientifically knowledgeable. And I think that scientifically knowledgeable really helps ground the recommendations and the discussions that we have here in agronomy. So everybody's familiar with this, right? So this is what we learn in 8th grade biology is you take sunlight, water, and carbon dioxide, some chlorophyll, that's photosynthesis, and makes carbohydrates and oxygen, right? So that's kind of the basic understanding of plants that we all get in middle school, right? So I think that while that is true, that is part of plants, maybe thinking of that as a small corner of what is actually going on in the plant is also very useful. So carbohydrates are not necessarily the only thing we need within our plant. What is going on? My mouse is freaking out. There we go. So I like this a little bit better. So it really is a cycle, right? So we have sunlight, energy, water, carbon dioxide, photosynthesis makes carbohydrates. But carbohydrates are then used to make amino acids. Amino acids make proteins. Proteins make plants, and then plants continue this cycle, right? So we grow a stronger plant with more plant or whatever it might be, and we can have more photosynthesis. Now, there's also a lot of other inputs that go on here. We have the reason we apply fertilizers in general is because we need additional nutrients to handle other parts of the crop metabolism, right? We also have this production of antioxidants, which we'll talk about here at length. So that's increasing the complication here. We never really talk about how the plant decides what part of that plant needs to grow, right? So there's lots of different things that plant needs to produce. Not only is it carbohydrates and amino acids, which have lipids, as well as a whole host of what we call secondary metabolic pathways. These are these terpenes, the phenolic compounds, the antioxidants, the phytochemicals, the phenolic compounds, and so on and so forth that really drive the functionality within that plant to say, I'm gonna grow flowers, I'm gonna grow fruit, I'm gonna grow roots, I'm gonna grow more leaf material. And it's really a complicated interlocking system of chemistry that's going on within that plant. It really comes down to this well, it goes lower than this, but it really comes down to chemistry within our plant cell, right? So when we want to kind of frame the discussion around plant stress, it's really at the simplest level, it comes down to chemistry within the plant cell. So when we have chemistry going on within the plant cell, whether that's photosynthesis, that's a chemistry process with enzymes and things like that, or it's lipogenesis, making fats, or it's making DNA, or whatever it might be. It's a chemistry standpoint. It's plant respiration. There's chemistry going on. And when we have chemical reactions, we have lots of byproducts of those chemical reactions. Now, in a well-functioning plant cell, those reactions will be balanced, right? So we have the ability to produce a new carbohydrate, or a new ribosome, or a new piece of DNA, or whatever it might be. And there's byproducts, just like with any manufacturing process, whether we're manufacturing cars or toothbrushes or fertilizer, we have these byproducts, right? And that happens at the level of the cellular metabolism as well. And the problem there is when we start having stress impact the cellular metabolism, those reactions are less and less balanced. So we have more and more of these byproducts coming into the scene. You think of it more trash building up within the cell, right? And that trash isn't great, right? So the trash is actually reactive within the plant. So these are things that are going to oxidize other molecules within the plant and cause problems. So you can think of them as stress builds up, that we're going to have more and more of these oxidative compounds building up within the plant and within the cell. Now, what that looks like is, under prolonged stress, we're going to increase the amount of excessive oxidative compounds in there. Now, those will start reacting with things. They're not just going to sit and be nice. They're going to have problems. So you can think of these little chunks of chemical that are going to be running around and trying to react with something. Where usually in a properly functioning cell that's not under stress, we will have it'll be taken care of. There'll be a balanced equation there. So what the problem is when stress continues and gets worse, we start seeing more and more of these oxidative compounds show up, and they start causing more and more damage, to a point where eventually we can have cell death, decreased respiration, all kinds of issues. And of course, it's a feedback loop, a negative feedback loop, where if you get a little bit of stress and then you get more stress, and it's harder and harder to recover from that stress. And that's where we start seeing major issues within our cropping system. So this is what we call stress. So a lot of this is abiotic stress. So abiotic stress is the negative impact of non-living factors on plant growth, yield, and quality. Abiotic stress will reduce photosynthetic activity, plant respiration, cell wall strength, and root growth and metabolism. So I think we talked a lot about what some of these stressors might be, and how we kind of work to manage them in our first half of our talk of this hour. But I wanted to go through a little bit here and just kind of highlight some specific stresses and what they directly do to the plant. So I want to start with some weather stresses. We talked a lot about wind, extreme temperatures, as well as moisture problems. And so heat and cold can really reduce our respiration. And we'll go through a specific case example here in a little bit of a heat stress and what that looks like within the plant. Lack of sunlight, that was brought up by John, I believe, that when we have poor sun conditions, that really can really impact our photosynthetic potential that we have within that plant. There's less photons, photosynthesis is going to run a little slower. Soil moisture is a big one, and root metabolism. Gordon mentioned that we have that phytophthora is a huge issue with and that's called avocado root rot, and that can be directly related to soil moisture issues. And then we also have humidity and precipitation can cause issues with antioxidant production and cell wall strength. Plant and soil nutrition obviously also plays a big role in how a crop deals with stress. Nitrogen, and all of these are true in both our toxic levels as well as our deficient levels. Bob brought up here in the chat that that boron can be one of those, can be a nutrient where it can come into a system and it can be extremely beneficial at certain times. But when we have excessive boron, it starts causing issues and toxicities. And we see that a lot of lemon and avocado production throughout kind of the Ventura County region. There's some areas that just have very high boron and can cause lots of problems. So in terms of nitrogen, we can get poor metabolism if we have too much or too little of it, and we'll talk about that a little bit more at length. Potassium, poor water, and nutrient assimilation can be a key role there. Calcium and silicon, when we're low there, we'll build weak cell walls, and that will lead to poor antioxidant production. As well as just in general, when we have inadequate plant nutrition, we can have poor root development, which then has a cascade of impacts. When we have poor roots, we can't pull up water, we can't metabolize properly, we can't store nutrition well. We are predisposed to having root stress of other natures. And the same thing if we have inadequate nutrition, we're going to see poor photosynthetic activity just due to lack of building blocks to even build the cellular mechanisms to do photosynthesis. If we're short on magnesium, we can't build chlorophyll because magnesium is a key part of that reaction. And then agronomic practices, sometimes it's our own fault that we cause stress, whether it's poor water management or phytotoxicity from an application that went wrong. That could reduce crop vigor. Soil management practices in general can develop poor root development, can induce poor root development, and so on. So abiotic stress has this cascade of impacts. Now, the reason we're talking about all this is because when a crop is under stress, that has specific impacts on how that crop can respond to other stressors. So abiotic stress will lead to reduced antioxidant production within that plant, which we just talked about. Antioxidants are very important just to keep the cellular mechanism functioning within that plant. The reduction in antioxidant production can lead to increased susceptibility to bacterial, fungal, insect, soil pathogen, and nematode damage. So we'll talk a little bit more about that here. So biotic impacts of stress. This is generally when we start, or this can be in some cases the first time that we start noticing the impacts of these stressors. So, for example, when we have lack of photosynthetic activity or antioxidant production, think cloudy conditions, moist cloudy conditions, that's putting stress on the plant. And when that plant is under stress, bacterial pathogens are going to have a lot better environment, not only climatically, but also within the plant to take hold and proliferate. Same thing for fungal pathogens, low respiration and antioxidant production. Arthropods love weak cell walls and low antioxidant production. Think a stressed plant. And then nematodes as well love a poor root development system with low antioxidant production. So obviously my background is in entomology. So I always think when I think arthropods, weak cells, I always think in plant stress, I always go to spider mites and strawberries because it's one of those systems where we have weak cell walls, but then we also have a heat event or dust or things like that on the leaf. Dust is a silly thing because you think of it, it's like it's just some dirt on a leaf. It's not a big deal. Why would that have anything to do with whether or not a mite likes to be there? Well, the mite likes that drier condition, that physically drier condition on the leaf surface, which is associated with the dust being present. But that dust is also stressing out that leaf, and that can cause a cascade of problems within the leaf, and that then produces an environment that the mite has an easier time consuming that leaf, and we'll talk about that here in a minute. I have a question here. Is it your thought that nems will attack Phytophthora impacted roots at a greater rate than healthy roots? So this is a question regarding nematode impact on roots, and yes, so a system where the roots are stressed, nematodes damage will be more impactful on the health of that plant. In many cases, we do a lot of work in actually turf as a good example here, where we can have really high nematode populations, and we can also have a very healthy looking turf situation because we have lots of roots down there underground. And if we have a healthy root system that's growing well, the damage that we'll see from nematodes will be inconsequential to the overall system. However, if that root system is not proliferating and growing and metabolizing and extracting nutrition, when we start losing those roots, we lose a substantial amount of functionality to that root system. So yes, so when we have Phytophthora present, the nematodes that are there will be able to hurt the roots more, and we will see a greater impact of that damage on the overall crop. If a, I have another question here from Patrick Troy. If a Salinas heat wave, high 70s, is forecasted 5 days from now, can a single shot of your product have an impact on lettuce stress and reduction? Let's put a pin in that one for now, and we'll come back to that in a little bit. So, but thank you, Patrick. We'll make sure we hit that here in a minute. So let's do an example here of high nitrogen stress. So plants need nitrogen, right? So they need nitrogen to grow, and evolutionarily, nitrogen has been a major limiting factor for plant growth. So if in the wild, if you're a plant and you're growing, nitrogen is the hardest thing for you to find in that soil. So over time, plants have developed mechanisms to preferentially take up nitrogen and store it. And a lot of that nitrogen is stored in a vacuole within the plant. So we'll have this vacuole that holds all of our nitrate or whatever nitrogen that we might have pulled up, in this case nitrate, and hold on to it. And so when we're holding on to our nitrogen, as we give that plant more nitrogen, the plant cell will fill up, and the plant will say, well, I need to hold on to this nitrogen because it's very important to me, so I need to make more cells. So it will scavenge nutrition from other areas of the plant to push more growth and be able to hold more of that nitrogen in the plant. That's why we see this. If you think about when we do a big nitrogen application, we see what we call, and sometimes rank growth or soft growth, that'll come out after a nitrogen application that's been maybe a little bit on the heavy side. Sometimes we want that, sometimes it's good, and other times it's not, and we're going to talk about that. So the problem being is when we're growing out that additional tissue, in many cases we are short on other nutrition, and that will lead to a weaker cell wall that's produced. Now that weaker cell wall is more likely to lose water under stress, is more likely to have just poor structural stability overall. And one of the biggest kind of ways that we see that impacting our production systems and our decision making is we see that the crop will have a lot higher susceptibility to damage from not only insects but also pathogens. In this case, we have a fungal spore here on a leaf cuticle. The cuticle is thin, the cell walls are weak, and this fungal spore has an easy time penetrating the cuticle as well as the cell wall and getting into the plant. Now the plant has natural defensive mechanisms that this is a battle, right? This is the battle between the plants and the funguses and these parasites, the fungal spores. So the plant has cell walls. They're there to stop fungal spores from being able to pull nutrition out of their cells. The leaf cuticle is there. One of its functions is to stop pathogens from penetrating into the leaf and accessing leaf tissue and sucking energy out of the plant, right? So what the plant does not like this, obviously, and is developed mechanisms to fight against having parasites on there eating it. So this type of situation is made worse when we have these soft cells. The fungal spores are able to access them easier. Now, interesting enough, fungal spores are also nitrogen limited. That's the hardest thing that they have to find. So they will grow better and faster on a plant that is high in excess of nitrogen, and then the fungal population will grow faster as well. This is also true for insects. Did you ever wonder why aphids or mealybugs or something like that, why they pee out all that sugar? Because we always think sugar should be a great nutrition source for plants, and that's somewhat true, but they also need they need protein and nitrogen, specifically nitrogen from that protein. And so they're actually filtering out the phloem or the xylem liquid to get access to more nitrogen. So when we have more nitrogen present within the system, they will grow faster and be a stronger population to control. So the plant does all kinds of things to help fight against these types of things. They have these secondary metabolic pathways to form defensive compounds that help the plant resist these types of stresses. They also build thicker cuticles, stronger cell walls, and things like that. And some of this is induced under under parasitism from some type of stress, and some of this is just inherently present within the plant. So it really, I think, comes down to cell walls. So let's talk heat stress. So getting to Patrick's question here, heat stress. So sunlight energy is obviously very important, but when we get too much sunlight energy, it can also be a problem. And we get too much heat that comes with that sunlight energy, that can also be a problem. So I wanted to throw up this picture of this last year. We've had 2 years in a row here of extreme heat stress on the coast. Here's my daughter holding our 116 degrees that we had, and actually went to 120 that day. And I took these pictures with a heat camera, and you can see that the fruit was 104 degrees on the surface, and the leaves were 108 degrees with ambient pushing 114 at this ranch, and we saw 122 at another ranch. So what happens when this leaf goes to critical temperatures, right? So how does that influence? So just like any other machine or system, it's going to start shutting down. And so that actually happened to me when we were driving around at 120 up a mountain looking at avocados, because that's what you do when it gets 120. You go look and see how things are going. And the first thing it did was it said, this is my truck here. It says power reduced to lower engine temperatures, right? So we're overheating. So the plant will do the same thing. It'll say, okay, it's too hot. Let's try and push more water through the system to try and cool down. We're going to slow down our internal mechanisms, but we're going to try and move more water. Well, at a certain point, we can't move enough water and we're still too hot. So that's when the leaf goes into survival mode, which my truck also did. And so at 175, apparently in your transmission or your transmission in your motor, it gets, or 275, it also gets angry and it tells you can't drive anymore. So luckily everything was fine. It cooled off very quickly. The truck, I made it back down the mountain. It was no problem. But you shouldn't drive up mountains when it's 120 degrees in an F-150 apparently. So the plant does the same thing, right? It'll shut down the system to try and conserve water and go into survival mode. It closes off the stomata to keep water in the plant, and then it also will try and slow down the rest of its metabolism and just try to hold on for dear life, right? So we talked about this. When the leaf is shut down, we want to make sure that it keeps as much water as it can. If we have poor cuticle development, that crop is going to lose more water through other means other than the stomates. It'll go through the cuticle and so on and so forth. And then we'll also we'll just lose time and photosynthesis activity. And like we talked about, antioxidants build up and we see cellular damage over time, and this can cause problems. Now, our goal being, we want to build a crop that is able to manage these oxidative compounds, not lose energy through cellular strength, and be able to maintain a strong cell wall. So and then respond appropriately through plant respiration. So overall, our goals here are build a strong cell, make sure our plant can respire appropriately in the conditions it has, make sure our photosynthetic activity continues under stress, because photosynthetic activity directly relates to yield, which is our goal as farmers, is to hit a yield goal. And then overall, I think we'd like to talk about kind of how can we do this from input selection. So that's what I'll talk about in the second half of the hour, this next hour. But that's the end of this section, kind of on how does stress physiologically impact the plant. And we'll come back to a lot of these concepts here in a little bit when we discuss kind of the Redox stress defense framework. So with that, I'd like to introduce our next speaker. Our keynote speaker is an IPM expert focusing on utilizing IPM theory to solve real-world problems. As the entomology and biologicals advisor, he primarily looks after pest management along with research on biostimulants, biopesticides, and other biological materials. He has authored 370 scientific and extension publications that include 19 book chapters, and is the subject editor for the Journal of Economic Entomology. His current focus has been redefining IPM through the lens of a modern production system. His talk today is entitled IPM and Managing Plant Stress, How Growing Healthy Plants Fits into the IPM Model. It's my pleasure to introduce Dr. Surendra Dara.
Good morning, Danny. Thank you very much for inviting me to participate in this webinar. You all can see the screen well?
Yeah, I see your PowerPoint.
Okay, very good, very good.
We're good.
So thank you. And it is great that the panelists and Danny gave you a very good overview about the practical experiences about stress and then the technical aspects of plant stress as well. So it makes it very easy for me to go over some of the details in my presentation. So one more thing is like, can you see the pointer or no?
Yes, I can see the pointer.
Okay, very good. All right. So, so what we see here is the IPM model, and the focus is pest management, not looking at anything else, and how we manage pests or integrated pest management is through emphasizing on cultural control aspects. So, you know, the water management and nutrient management and all of them are related to cultural control, and the focus is that one. So if you look at the plants and nutrition, basically when you have a weaker plant or undernourished plant, you have impaired growth because it is under stress, not growing well. And because it is weak, it is more vulnerable to various stressors. And as a result of that, you have reduced yields. When you have overnourishment, like excessive nutrition, the plant might be growing bigger and looks greener and looks nice, but there could be a lot of physiological changes that are impacting or negatively impacting the overall production here. One, you have excessive vegetative growth. Sometimes you know, it can, the resources are directed towards the vegetative growth than the reproduction of fruits or vegetable parts that we want to increase. And then it also attracts the biotic stressors. When you have excessive nitrogen and sometimes P and K, what happens is it increases the carbohydrate content of the plants and reduces the anti-stress compounds like phenols and other materials will be less, and carbohydrate concentration is higher. Because of that, plants become more attractive to pests and diseases. And then additionally, you might see increased yields to some extent. In some cases, you might see increased yields, but because plants have become more attractive to these stressors, you might also end up applying more pesticides. So as a result of that, you might end up having lesser returns. And you should also look at this negative impact of any of these practices, like increased fertilizers can leach and contaminate the environment, and increased attractiveness to the pests is leading to excessive pesticide applications, and it has its own negative impact. So when you have an overnourished plant situation, you might experience one or more of these consequences. So when you have this optimally nourished plant, you know, the growth is optimal, yields are optimal, health is optimal, and yields and returns are also optimal. So that is our goal. So making sure that plant is receiving enough nutrition, water, and all the care it requires, so that it can we can maximize the yield potential. Now, how healthy plants and you know, IPM come into play. So here, just explaining this whole thing again, when we have healthier plants, they are less attractive and less susceptible to pests. And then even if there are some pests, which is going to happen, or some unfavorable conditions like temperature extremities or salt and other stress factors, then they can withstand those situations better than a healthy plant. Then when you have optimal yield and health, you know, optimal growth, higher yields, it can compensate for any losses you may have from these stress factors. And additionally, it reduces the reliance on pesticide applications or any other practices that are necessary to bring them back to that optimal condition. So if you maintain it right from the beginning, we can avoid some of those. So and finally, it minimizes the risks associated with the excessive pesticide or fertilizer applications, and and also the cost associated with it. So when we are trying to maintain good health of the crop, it has a lot of benefits. And how do we do that? Choosing, this is all about cultural practices. How do we ensure that the plant is growing optimally? Like choosing the optimal season, plant density, and soil conditions, which seems redundant to mention. These are like generally well taken care of. Nobody's usually growing them out of the season and in unfavorable conditions, but those are the primary, these are the basic principles of cultural control. And then optimal irrigation and nutrition. There has been enough said in the previous talk and discussions, so we understand why it is very important. Then improving soil health. This is also very important because the soil is a very dynamic system. It is not just the plant roots that are growing there and some harmful arthropods or pathogens that we have, but it is also full of organic matter and lot of beneficial microbes and minerals and so on. So it is a very dynamic system, and what you have in the soil is going to govern the whole plant's performance below and above ground. So it is very important to maintain soil health, not just for optimal plant growth, but also for good environmental impact in the long run. Then regularly monitoring and addressing these stress issues, whether it could be an entire field-wide stress issue or maybe in isolated location. So we need to be regularly monitoring and take care of those. So and finally, sanitation is also very important. If there is a diseased or a pest infested plant or some plant debris, we want to remove that so that they don't act as source for further spread of the infection or infestation. So these are all cultural practices that broadly we can mention for maintaining good crop health. And the focus of this presentation is just nutrition and soil health. I'm not going to go into the details of the various mechanisms, just to give you, I'm going to give a general overview here. So plant biostimulants, I just want to define what biostimulants are first. And this is just my definition, and you might see various versions online or in books. So these are basically microorganisms or microbial derivatives, or they could be of mineral, marine, or botanical origin. And what they do is basically stimulate plant physiological responses and induce systemic resistance. And as a result of that, they increase the plant, nutrient and water uptake, promote plant growth and health, and impart tolerance to biotic and abiotic stressors. This is all without having any pesticidal properties, basically. It is all related to stimulation of biological processes, but not directly having any nutritional value or pesticidal properties. But there could be slight deviations to these definitions, because some of the active ingredients used as biostimulants can be pesticides too, depending on the rate, depending on the strain, and so on. And some of these products also come with some nutrients. It is not the active ingredient that provides the nutrition, but in the process you have other materials too. So they also provide nutrition, but our focus is what are plant biostimulants as various categories that I'm going to explain and how they can impact. So these are the categories. We have 9 of them. So these are arbuscular mycorrhizae. These are some examples: Glomus, Jagospora, and Paraglomus. These are the genera and different species within them. And then you have entomopathogenic and other fungi. So trichoderma, as you see here, trichoderma is something like many of you are familiar with, but Beauveria bassiana and Metarhizium, which are primarily used as the biopesticide ingredients, they can also have this biostimulant impact. And then you have plant growth promoting rhizobacteria, several species of them, and then protein hydrolysates. And these are of, when you hydrolyze these animal or plant-based materials, you get this protein material, and they have an impact on plant growth and health. And then humic substances. These are basically biologically derived organic matter, and they are they are a major source of organic matter in soil. And humic acids are, one of the examples that we are familiar with. And then algal extracts. There are those based on microalgae and those based on macroalgae. Macroalgae are also known as seaweeds. So you're familiar with both these categories. And then botanical extracts. Then again, here we have several examples of black walnut, dandelion, garlic, neem, and soybean extracts. And some are used as biostimulants, and you're also familiar that some could be used as pesticides. But the issue here with some of these active ingredients is that they have multiple roles, and we are focusing on one of those. And then silicates and phosphites, and they are also available in multiple forms as silicates are phosphites of sodium, potassium, and calcium, and so on. And Danny explained why it is important to use some of these materials to strengthen the cell, the tissues. And then finally, we have chitosan. Chitosan is obtained from the exoskeleton of insects and shellfish. It is a part of chitin, so it is a polysaccharide, and it can be again a biostimulant or biopesticide or biofungicide. And if you look at algal extracts, here too they have a lot of polysaccharides that have similar properties that other materials have. So these are the 9 categories of plant biostimulants that we have. And how they work is what I'm going to explain now. So the first thing is they induce systemic resistance. And I will explain that in the next slide. But then they also build soil structure. When you add these, you are increasing the organic matter content and soil water holding capacity, and then the nutrient cycling. And because of this organic matter, you also have a lot of beneficial microbial activity, and which again in turn influence the plant health and the soil health, and they can antagonize plant pathogens and so on. And just like I said, and then regulating various biological processes that attract beneficial arthropods or beneficial microbes as well, because plants communicate through chemical signals. So whenever they are exposed to some stress, they have this chemical network activated and send out signals, alerting other plants and also attracting beneficial. So when you have a plant and you apply, one or more of these biostimulant products, and what happens is they trigger this biological processes, and then also send the signal out to attract these beneficials. So this is directly, they are helping plants become stronger, and indirectly, they're also attracting enhancing biocontrol or microbial control. Then briefly about induced resistance in plants. So when plants are exposed to these arthropods or pathogens or sometimes chemicals and these stress factors, they trigger this salicylic acid pathway and also produce pathogenesis related proteins. So these are basically proteins that help plants fight the infection or any of this herbivore damage. So this is called systemic acquired resistance. But the other kind is when you expose plants to beneficial microbes and some of these biostimulant materials. So what they do is they trigger this jasmonic acid and ethylene pathways and also help plants produce these pathogenesis related proteins. So this one is also a defense mechanism. Both of them are activating certain sets of genes, sometimes the same sets. They share, some of these genes are involved in both kinds of pathways, but this is the other kind is induced systemic resistance. So the first one is when plants are exposed to stress, so it is a reactive mechanism. And then when we apply these materials, it is a proactive mechanism. Many growers, knowing that there are going to be certain pests and diseases, apply pesticides proactively. So which is what we are proposing here, or the science is proposing here, is instead of spraying those chemicals, why don't we try to be proactive in terms of adding these beneficial microbes or other biostimulant materials, so that you're preparing plant not just to control the pests but also to grow well and be stronger and so on. When we are spraying pesticides, it has a specific activity. It is going to be active against the target pests or disease. It is only going to kill it, and it might also kill beneficials. But when we are applying these beneficial microbes or other biostimulants, it does not have those kinds of negative impacts. It has multiple purposes, as I'm going to explain in a little bit. So this is what induced systemic resistance is, and that is the basis for using biostimulants. And then these are various categories of biostimulants, like I mentioned earlier, arbuscular, mycorrhizae to chitosan. And how you use them is you can treat the seed, or you can treat the transplant, or you can apply to the soil or to the foliage throughout the production season, depending on your need and what the active ingredient is supposed to do. And then next is what does it do? When you are applying either to foliage or the soil, the first thing is that it is inducing that systemic resistance. If we are talking about just the plant itself. And if we are talking about the soil, then it improves the soil structure and increases the beneficial microbial activity, and also it induces the systemic resistance. So one example I quickly want to mention is that the plant roots, the exudates from these roots they stimulate these beneficial microbial activity, because the plant, when knows, just like it is sending a signal out for these predators or parasitoids, it is also sending a signal through root exudates to these beneficial microbes. So when we add some of these biostimulants, then the whole mechanism, the physiological mechanism, or the genes are going to be activated, and they communicate with each other and change the plant chemistry and attracting these beneficials, and which directly or indirectly suppress these biotic or abiotic stresses. So when we have this, what happens in plants is that it increases the photosynthesis. Then it also increases the antioxidant enzyme activity, which can fight these stressors. And then it also produces volatile organic compounds and strengthens the plant tissues and improves nutrient and water use, uptake, and mobilization within the plant, and increases the stress tolerance. And what this results in is increased root and shoot growth, and then increased yields, quality, because of all these antioxidant materials, and then also increases the shelf life. It is not just when the plant is growing, but the impact of these biostimulants can last even after harvest. And then it increases the attractiveness to biocontrol agents, as I mentioned, and then also beneficial microbial activity. And as a result of that, it reduces the pest and disease levels and their damage, because they triggered mechanisms that suppress them. And finally, we have optimized use of water and nutrients. So there is some saving there. And then optimized use of pesticides, and then reduce risk of pesticide resistance, because we can save them for real serious situations, like the minor infections or infestations can be controlled or addressed through these other non-pesticidal activities. And then improved biological and microbial control in the entire system, and the ability to grow crops under unfavorable conditions too. If even in these arbuscular mycorrhizae, some of them are very useful in growing crops in under drought conditions or saline conditions. Then as a result of this, we have improved environmental health as well, and then optimized production costs and increased returns. So with these kind of biostimulants and biologicals, we can effectively use them as a part of cultural control of IPM and contribute to sustainable agriculture. And this presentation is actually a part of my chapter here that is advances in biostimulants as an IPM tool in horticulture. This is a chapter in this upcoming book. It has a lot of other topics related to basically IPM in horticulture. If you are interested, you might purchase this book when it is available in November. And this is the end of my talk, and you can see my other presentations online at my YouTube channel. And with this, I end my presentation, and if you have any questions, I will be happy to answer.
Ranger, you've got a couple in the chat there. Would you like me to read them to you from Eduardo?
I'll check that. I'm going to stop sharing my presentation first, and then I'll see this. Okay. Okay, Eduardo is saying that I guess that proactive strategy by applying beneficial microbes has a ceiling of efficacy dependent on pest pressure. Has that been measured or modeled? As I mentioned, the soil is very dynamic. It all depends on what kind of microbes you're applying and what is there in the soil, whether the soil conditions are going to be ideal for that, and the crop, soil, temperature, organic matter, and so on. In general, we want a diversity in those microbes, right? Like it is not just one microbe, but when there is a balance, each one compensates for the deficiencies or other inadequacies from other ones. So to answer your question, if there is a ceiling, I am not quite sure. It all depends on the stress the plant is going through. If there is a pathogen, or if there is an expected stress, maybe you will see the benefit, but if there is no stress, you may not see any benefit. But as Danny was explaining earlier, what happens inside the plant is more complex than we can see. So even if you don't see the benefit directly in terms of increased yield, it might be addressing that heat stress or salt stress or some other situations that we may not have seen. So it is difficult to see, but just like anything, there is going to be a ceiling. You can only apply so much. The ceiling could be related to the environmental efficacy or the cost. Yeah, I can apply a ton of this material, but it is too expensive, then that automatically sets the ceiling. I hope that helps to address your question. And then the next one is Tracy's asking, reduce his own screen size so the slides are larger. Okay.
I think he took care of that one. You're good now.
Yes, yeah.
There's one more above Eduardo. Eduardo had another question above that first one.
Okay. Considering that the susceptibility of plants to a given pest level pressure is influenced by plant health, how should classical decision-making protocols or tools like economic threshold models be changed according to plant health conditions? So that is actually a great question. These economic thresholds are you know, I mean, it takes so much effort to set these thresholds, but they can be dynamic too. They can be changing from season to season and depending on what many factors. So you're asking a question, should it be changed or what is the impact? Yeah, it can change, but it is very difficult um, because it requires a lot of studies and a lot of input from various fields and from from a diverse conditions to set those. Yeah, I agree with you that you know, your point basically, yeah, the thresholds could be changing frequently, but they don't, because we don't have the resources to change them. And are there other questions? Does carboxylic acid has biostimulant effect on plants? Um, yes, it is. It is. It has an impact, biostimulant impact on plant in the sense like they can be attracting these beneficial microbes, carboxylic acids. That is one of those biostimulatory effect, and it also has an impact. I'm not an expert in this area. I am an entomologist like Danny, but you know, I do research with these to find the effect in the field. I don't get to look at all the molecular level interactions and everything else that happens within the plant. But I'm sure that others may have an answer for that question. Any trial work with biostimulants and effects on Pythium root rot in Salinas Valley lettuce? No, I haven't done that yet. No, I haven't done that on Pythium. But I have done some studies using entomopathogenic fungi for suppressing strawberry soil-borne diseases in strawberry and cotton, like fusarium and macrophomina and so on. So there was some positive impact from those studies. Then Troy is asking, in my opinion, does adding organic based products or products with carbon to nitrogen ratios that are beneficial to soil biology help in conventional systems? I would think so. If even in, you know, organic matter is important in any system. And if a conventional system is not focused on building the soil structure with organic matter, then there could be some negative impacts. With the other efforts of adding fertilizers or pesticides, we might be able to take control of some of the issues and not realizing the benefits. But soil health is not just for a short-term benefit. It is going to be a long-term benefit. And I actually saw something on news this morning. You know, someone was talking about how fungi are important in the soil for the soil health. Uh, they are a huge part. We are talking about beneficial fungi. They're a huge part, and not necessarily trichoderma kind of thing. Those that are just in the soil doing their mining their business. They are huge carbon sink. So they are going to, whether it is dead or alive, it is going to accumulate this carbon and increase the organic matter content and so on. So, you know, to answer your question, yes, it helps conventional systems too. These questions, I hope these are for everybody. I may not have answered everything as you would like to know. So Danny and others can also answer.
I've got one over here in the Q&A section. Does carboxylic acid have biosimilar effects? I think you already addressed that one. And then the follow-up is it better than humic or fulvic acid?
That is a tricky question. I'm not sure. In my reading, I think humic and fulvic acid are probably much stronger because of what I read recently when I was writing this book chapter. But you know, I cannot dismiss the impact of other ones. You know, everything has its role.
Can I add a thought on that one? Is carboxylic acid is an actual acid in terms of a chemical definition of an acid, correct? So it has its role, I would say that humic and fulvic are in a completely different kind of category, where they're more soluble carbon, larger complexes, especially compared to a carboxylic acid, which is a very small defined molecule chemical analysis. So the humic and fulvic, I think that those are not really comparable. It's apples to oranges. I think the debate around humic or fulvic acid, which one of those might be better, I think that could be debated. But I think comparing the two is comparing two categories that are kind of complementary to each other or independent of one another. Have one more here in Q&A. What in-field instrumentation can be used to measure stress or some of the plant responses you noted?
I think, field stresses like the ET and water level, and then, basically looking at the plant growth, the parameters, general parameters like photosynthesis and accumulation of nutrients, nitrogen and other nutrients in the plant tissues are some of those things that we can see. And ET is probably another major factor telling us how it is the plants are absorbing water and growing. Maybe the agronomist can answer this question better.
I think that functionally, there's several different technologies on the market depending on your crop that can measure kind of water status of the crop and things like that, depending on your stress. The problem with measuring stress in a general format is it's a pretty massive field of kind of metrics you're trying to look at there. So there's NDVI, there's all kinds of other kind of visual spectrums you can look at, and obviously just going out and looking at your crop, you can always look at it and kind of see some notes of stress. So it's the problem I see with a lot of these instruments is what degree of stress can they measure versus what you can already detect just from being present. So there's a lot of trade-offs in terms of tools versus presence in the field. Okay. Dr. Dara, thank you so much. I think that was a very enlightening talk, and I look forward to reading the whole book chapter in a couple months when it comes out. So thank you.
Thank you.
If anybody has any questions, Dr. Dara, you may be able to hang on here for the next little bit, and if more questions come up, we can address them at the end.
Yes. Yes.
Great.
Yes.
Thank you. Okay. Let's go back here. Okay, so building on kind of everything we've talked about up to this point, obviously this is a Redox sponsored webinar. So let's talk a little bit about what Redox can bring to the marketplace of plant stress and kind of the framework we can use to address abiotic stress defense. So Redox is a bionutrient company that focuses on sustainable plant nutrition. That's what we do. We went over this earlier at length of kind of how abiotic stress influences the plant and the cascade of impacts it can have when our crop is under abiotic stress and what it can do to the plant. There's really four pillars of that we like to rely on for improving the crop's ability to deal with stress. That's fortifying cell wall strength, accelerate photosynthetic activity, increase plant respiration, and then stimulate root growth and metabolism. We want to fortify cell wall strength for this purpose. We have more when we have a better cell wall. That plant will be able to perform its normal metabolism better. You can think of it as a better foundation for everything that goes on within that plant. It's also going to be able to deal with stress as it comes up in the field. And as we talked about earlier, when we have a strong cell wall, a strong cuticle, and so on and so forth, it's less likely that we'll have damage to that crop from external sources, including biotic stressors. We want to make sure that we have strong photosynthetic activity within that plant so that we can produce the needed molecular products that we need to produce a strong crop. We're in the business of producing yield, and whatever that harvestable yield might be, whether that's a head of lettuce or an almond or an avocado or a strawberry, But all of that comes down to the overall strength of that plant and its ability to push resources into that harvestable unit of that crop. So not only I hear it occasionally, I hear it less now than I did when I first started this job, but I hear it occasionally. We don't sell lettuce roots, right? Well, that's true. You don't sell lettuce roots, but you do sell lettuce heads, and that lettuce head is directly connected to those roots, and those roots make sure that that lettuce head weighs enough to end up in a box. So we want to make sure that we have a crop that is able to have cellular respiration as well as photosynthesis that is able to achieve our goals. And lastly, we want to make sure that we have a crop that is able to stay in that balance that we talked about earlier. We want to make sure that oxidative stress is able to be avoided or reduced, and increased ability to withstand external and internal stresses. So that's really our goal in this fortifying cell wall strength, accelerating photosynthetic activity, increasing plant respiration, and stimulating root growth. Now, in your packet, you got this little booklet that's about abiotic stress defense product guide. It goes through the products that we're going to talk about now, and yeah. So if you want to make notes in there, there's some basic information on each one of these products, so you have that. If you didn't sign up early enough to get a physical packet, you got one in the email that you should have received yesterday, and we'll be sure to include that in the follow-up email we'll send you guys soon. So after this, so let's start off here with our first tactic. So this is Mainstay SI. It fortifies cell wall strength. It's a calcium and silicon product, utilizing our microencapsulation technology to make sure that we maintain the availability of that calcium and silicon and get it into the plant. Really, the root here is increasing antioxidant production at the plant as well as fortifying that cell wall strength. This tool is also available in an organic formulation. The following tools we'll be talking about aren't available in organics, but this one is. So if you are an organic grower, have organic production under your umbrella, Mainstay SI is really a cornerstone of a lot of these abiotic stress defense programs in building a stronger plant, right? So I did a lot of work with silicon over the years, or I have done a lot of work with silicon over the years, and being able to have a crop that is structurally sound is really the foundation to a lot of this abiotic stress programs. So the goal here of this is, we talked at length about how when we have weak cell walls, we have a weak cuticle. We'll lose water through that under heat events. We'll also will not be able to function as well under stress. When we add calcium into this system, it gets much better. We have a improved cell wall structure and better organization of those cell walls. We have a better cuticle that's developed. Overall, the plant cell will function better, and we see this a lot in just increased quality of that plant over a system that doesn't have added calcium into it during growth periods. And then when we add silicon in, we see that amplified, where we see calcium having a much stronger impact, enhancing cell wall structure. The silicon is extremely helpful in building that cuticle as well as those other leaf structures and maintaining the rigidity of those cells. So that's really what the goal of having silicon in the system is for. And we see that a lot here. So I want to tell you just a couple stories about where we found fits with this in a couple different production systems. So strawberries here, strawberries are a soft fruit, right? In terms of a general characterization, it has a host of issues with how fruit can be culled out, and it has a high fresh picked strawberry market has a very high standard of quality. Right. So there's a lot of different ways we can lose fruit. Here's two options here for some pathogens taking out some fruit. And I was standing there with a friend that's also in the strawberry business when I first started with Redox. We were talking about how we have this Mainstay SI product. It seems pretty cool. I did a lot of work with other silicon products during my PhD, including a little bit with Mainstay SI. And we're talking about, I wonder how much impact this could really have as just a standalone. What do we do if we just took this product and added it into a system? So luckily he was in a position where that was his job, was to see what tools worked in strawberries. So we kind of put together a protocol and we did a project where we looked at how mainstay SI affected coal rate within strawberries, and did a pretty basic program of mainstay SI applied at a half gallon every 14 days as they could over the winter. It happened to be a very wet winter that year, and so there was lots of reasons to cull strawberries out, and they just looked at total yield and marketable yield and some cull rates. And what we found is when we added mainstay SI into the system, we were able to increase total yield by about 7%. So that just means we got overall more fruit set on the crop, bigger fruit, better crop overall. Now the impressive thing was our marketable yield increased by 10%. So that, sorry, 11.5%. So that means that we culled less fruit when we had it in the system. So overall, we got an 11% bump in yield with adding mainstay SI to their system. This was a standard strawberry production system that they did this trial in. So that was a very nice improvement to see. So that was fun. So the funny thing was both he and I are entomologists by training. And so we wanted to see, okay, well, that's fun, but we like bugs. So what happens with bugs? Now, we did several projects together, and I ended up working with kind of this concept here, and this was the overarching concept. Well, if we're improving fruit quality, that might mean we're increasing the skin integrity, right? And that would correlate to penetration force of that skin. There's a lot of research that shows that for this pest, which is spotted-winged Drosophila, we make a thicker, tougher skin, that we should get less spotted-winged Drosophila. So that was an interesting concept, and we'd had some reports in the field from people that was true. I reached out to Mark Bolda up in Watsonville to take a look at this, and he was doing a project on some blackberries, and it worked out that he was able to fit this in and kind of see what it looked like on some blackberries. So that was nice. So he looked at SWD adults and larvae when he put mainstay SI into the program, and what he found was when he treated with mainstay SI, he did see a marked reduction in larvae that were present in the fruit, as well as he seemed to see a slight reduction in the adult presence, but definitely saw a marked reduction in larval in oviposition. So I mean that's the damage that SWD, spotted-winged Drosophila, does in berries, is it lays eggs in the fruit, and then there's maggots in your strawberries and your blackberries, and nobody likes that. So it's a big problem in a lot of soft fruits, and that was a pretty fun thing to see. Is the whole goal here was, if we can build a stronger, tougher plant cell, we'll have this cascade of impacts, across fruit, across the crop, from being able to deal with heat and cold stress better because of that stronger cell wall, down to having an environment that's less susceptible to pest damage, and just not as good of a host for pests. I have a question here from Eduardo. What fruit fly species did you try mainstay with? And that was with Drosophila suzuki'i, spotted-winged Drosophila. So, sorry, I'm supposed to hit live answer, and then, sorry, there we go. Okay, so that's mainstay SI. So I want to start with this one as kind of the foundation, the framework of fortifying cell wall strength, because that's really, it's a physical characteristic of the plant. Is if we can build a stronger cell wall, it has a cascade of impacts from, like I said, all the way down from and even into storability, right? That's another place where this is it doesn't stop functioning when you pick it. It keeps working. So the next cornerstone here that I wanted to talk about was, sorry, I got off the screen here. Was Oxycom calcium. So Oxycom accelerates photosynthetic activity. Now, this product, it really increases antioxidant production. That is what this product does extremely well. It's a unique product on the market. It builds photosynthetic, it increases antioxidant production. So accelerates photosynthetic activity, and I really hear it referred to by a lot of growers that use this as gets the plants kicked into gear, right? That's really what it's doing. Is it gets the plants going so they can grow and deal with stress better. It's got a nice nutritional label, but really the goal here is let's push those secondary metabolic pathways. Let's get that plant dealing with stress. I wanted to show this picture as kind of a prompt here. Talk about a place where Oxycom calcium has been extremely beneficial, and that's been in grape production where we have uneven bud break on the cordons. When there's stress within the crop, we'll see that those further out buds don't break as well, and we just have poorer growth towards the end of the cordon. We've seen we've seen with Oxycom calcium used in the system that we just get a more even break out of dormancy when we use Oxycom calcium. Can be used in that, in a very early foliar application when we have early emergence, to just kind of get the plant going. Like I said, increase antioxidant production and help the plant to grow. We see the same thing in apples. We do a lot of work with Oxycom in apples, where we'll see fire blight damage, where we need to maintain growth and help the tree deal with the stress that fire blight brings. And we see Oxycom really being beneficial there, being able to grow through that. So I had a question here: can you use Mainstay SI in cannabis? And we use a lot in cannabis, so we can talk more about that here towards the Q&A at the end, of kind of where that might fit in. The next cornerstone here is DiCap. Now, DiCap builds on Oxycom a little bit, in that we've kind of shifted away from a focus purely on, I want to build antioxidant production with that plant, into a product here that has a very nice P and K source, where we have 03150, but it also contains a proprietary amino acids and soluble carbon, and some of the components of Oxycom as well, to build this product that we call DiCap. It increases plant respiration and antioxidant production, and really plays a great role in improving stomatal conductance and, like I said, phosphorus and potassium nutrition. This is a great product. It's a foliar or soil applied product that pushes the plant to continue to metabolize, as well as utilizes many of our technologies that we use for increasing nutritional efficiency. So this is a product where we can maybe apply 3 to 5 pounds per month in, say, an avocado crop over the summer, and that would be a sufficient P and K source that we're able to continue to grow that crop and fill fruit, even under the extreme heat stress we can have in summer production avocados. And same thing's true for summer production strawberries, where we can use this as a primary P and K input and see nice inputs there. And that is due to the potassium and nitrogen, or sorry, the potassium and phosphorus that's in there, and the complexing technologies we use. But it's also due to the phenolic compound production that plant is having when DiCap is used within the system. Like you said, DiCap's really helping push that antioxidant component within the plant that's over here, as well as making sure that we have good P and K to complement our nitrogen input, so we have well-rounded amino acid production and crop production, so we can continue to push size. Potassium is also incredibly important in stomatal conductance and heat tolerance. So when we use DiCap and get an efficient potassium source into our cropping system with minimal salt load, we're able to build a plant that's able to deal with heat stress a whole lot better. I wanted to show you this. This was taken. We took this little video and took these pictures in September last year in Salinas. So September is towards the end of the season in Salinas. It's usually small fruit time. We're talking usually sticking 15 to 20 berries into a clamshell or something like that. That clamshell's probably got, I don't know, 11, 12 berries in it. So just really happy with it. DiCap was used extensively to produce this result, as well as Mainstay SI, as well as Oxycom. Actually, all three of the products we've talked about up to this time were used in this field. Mainstay SI was extremely helpful here for maintaining fruit quality. As you can see, they're letting those fruit go pretty long on the plant. That's usually due to labor issues, where they run a lot of the labor leaves. So they're usually short on labor come late season in Salinas. So being able to keep fruit from literally rotting on the plant is extremely beneficial. And then we have Oxycom and DiCap coming to push that size and make sure that the plant's able to deal with late season stress, as well as provide P and K to that plant. So that was just a really nice, really fun field to go look at in September. That was just really doing great. A lot of the times, like I just talked about in the strawberries, we like to stack some of these technologies. So in this case, we did this trial to look at kind of what does it look like when we stack building a stronger plant cell with a product like Mainstay SI, and then helping the plant increase respiration and increase antioxidant production with a product like DiCap. So we did this trial with David Holden in Somas several years ago, where we looked at an application of DiCap at 3 pounds per acre and mainstay SI at half a gallon per acre, used with our fungicide applications in head lettuce production. Like I said, with the goals of what are the relative benefits of reducing that abiotic stress by increasing antioxidant production, providing P and K, as well as building a tougher plant cell. And we looked at this, and the gauge we used to see what type of how much stress that plant was under was downy mildew presence within the field. So so downy mildew was present in the field in our untreated checks, as well as in our fungicide checks. What we saw, however, is that when we used Redox products, notably mainstay SI and DiCap in our programs with our fungicide applications, because the fungicide applications were still necessary to keep down our pathogen pressures, what we saw was incidence and severity were severely decreased below our control as well as our fungicide checks. So, and that's not because we were increasing the efficacy of we did increase the efficacy of the fungicide slightly, but that was because the plant was able to deal with the environmental conditions better, as well as was a less susceptible host to those pathogens. So, so it allowed the fungicide to do its job. So the goal there being, let's increase respiration as well as build that stronger cell wall. The last product I wanted to mention here in our abiotic stress defense portfolio is Redox. So Redox increases antioxidant production. It's a botanical extract that stimulates root growth and metabolism. Now, this is really a lights out soil applied biostimulant for helping the plant deal with soil stressors, whether that's soils are too wet, poor root quality, whatever it might be. Redox has been extremely beneficial in building out root systems and helping the crop deal with vascular stress from below ground. We use this in a lot of situations where we have root stress and secondary infection from nematodes or pathogens. Redox has been very beneficial complementing those systems where we have a fungicide come in to help deal with a pathogen issue, but we still need to grow roots. So we have Redox there to help push that root growth as the pesticides will come in and take care of maybe that biotic stress that's holding our root growth back. So Redox is all about building that secondary metabolic pathway and helping that plant deal with stress through the production of those phenolic compounds. A lot of the times when we start talking about Redox, the question of, well, I already use seaweed inputs, I already use some type of kelp extract. What are we going to do? What are we going to do here? Do I put them together? Is it the same? Is it different? I don't understand. It's a botanical extract, so is kelp kind of. So where do we go? So that question came along. So we put together this trial looking at just a head-to-head comparison of Redox versus seaweed inputs, right? And we did this in celery. Celery is a great study crop because it's a pretty simple system. Plant size is the metric that we want. So and they have nice root systems that we can look at and things like that. So we looked at it pretty much just jumping to the answer here. We did them at the same cost, at the same rates, at the same timings, on the same fertility programs, on the same crop, same time, replicated. And what we saw was that over the grower standard, the seaweed product that we put increased by 18%. So that's a very respectable yield gain, right? 18%. We saw, however, that Redox increased yield by about 37%. So we were, I guess those results speak for themselves. The product has been one of the few products that I've seen in the industry where we run a trial and it consistently returns results over the grower standard program when added in. So it's been a nice product there. So really a cornerstone of growing a stronger root system is Redox. So here's our 4 products that we just reviewed. Mainstay SI to fortify cell wall strength, Oxycom calcium to accelerate photosynthetic activity, DiCap to increase plant respiration, and Redox stimulating root growth and metabolism. The benefits here of including these into a system is the crop is less impacted by extreme weather events. Yield is more consistent over time, reduced frequency of pest management applications, and then harvest quality and storability increases. Those are just a few of the notable benefits I wanted to highlight here on how these products can be beneficial within a management system. Obviously, there's nuance to where, when, and how, and what crop, and so on. And this is a very generic talk that covers dozens of cropping systems, so we couldn't really get into too many specifics of what time and where, but I hope you'll take a look at these and see where they might fit into your system. So that's what I have for today. I appreciate your guys' time on here and what we've done at this point. If you have questions, I'd be happy to answer them. I've already got a couple queued up here. So I'm going to start with these, and then if you have others, please shoot them over. I got to pull back up my chat box here. Sorry, the Zoom kind of moves things around for you. Okay, so I'm going to start with this one from Patrick that I stuck a pin in. So let's pull the pin out. So if it's Salinas heat wave, let's high 70s, or let's say low 90s, because that's happened a couple times the last couple of years, is forecast 5 days from now. Can a single shot of your product have an impact on lettuce stress reduction? And we've seen that that DICAP has been very usefully applied ahead of heat events. There is, I think, I'm trying to remember, somebody earlier mentioned there's a physiological ceiling to a plant's ability to deal with stress, right? So what we're talking about here is allowing that plant to deal with that stress slightly better. However, when it's 120 degrees, it doesn't work, right? That has surpassed the ability of the plant to deal with it. But we have absolutely seen that in Salinas, where if DICAP is used in a program or as a single curative approach or preventative application ahead of a heat event, where we'll be able to deal with it. 5 days ahead, yeah, we like to see 3 to 5 days ahead of a heat event, get out 2 to 3 pounds of DICAP as a foliar application. Can be very helpful to just help the plants deal with that heat stress better. Then we had another question. I keep forgetting to click this answer button. Another question was, can you use Mainstay SI in cannabis? We use Mainstay SI. It's actually our #1 product in cannabis. Cannabis is a silicon accumulator, which very few of our cropping systems are kind of classified in that category. Most of our cropping systems benefit from Mainstay SI. However, cannabis absolutely loves Mainstay SI. We get better structural integrity of the plant, as well as better leaf texture and things like that. So absolutely, Mainstay SI is used in cannabis. We'll use it as foliar applications throughout the production system, as well as substrate or soil applied throughout the production system. Next question from Josh. Can you explain how microencapsulation improves nutrient availability to the plant as a foliar, and soil application? So yeah, absolutely. So let's talk about that. So one of our problems with delivering nutrition to the plant is most of the fertility products that we have, or the nutrients that we want to provide to the plant, have some type of charge associated with them. The problem with that is they're reactive, right? So calcium and silicon are actually two great examples of reactive elements. If anybody's ever mixed calcium nitrate with a phosphorus source, you've experienced the beauty of how reactive calcium can be, and it ties up very quickly and it makes it hard to get things into the plant. So our microencapsulation technology helps to stabilize that charge. Calcium and silicon are both very hard to say chelate with a classical chelating agent like EDTA due to their size and their charge, their physical chemistry, charge structure, I guess we'll call it. So we've developed this technology that we call microencapsulation, which allows us to hold the charge on these larger molecules and be able to deliver them to the plant in a plant available form, but yet a stabilized form. So there's a tightrope there that we're walking, and these products do it very well. It functions foliarly or soil apply, so it maintains that availability, and we've seen it work phenomenally well in both applications. It also means that we can tank mix these products with other things, and it goes fine. For example, mainstay SI, which is 10% calcium, can be mixed with phosphite products for application with agitation, with no fear of no issues of clogging heads and things like that. So great question, Josh. Thank you. Eduardo says, I would like to have more details about the use of mainstay SI for protecting small fruits from spotted wing Drosophila. How can I get it? We've done the trial we talked about. There is some more details in there in your packets, as well as we can direct you to some of the other research that's been done with mainstay SI in small fruits. We have a couple of good ones in blueberries and other areas. If you want to reach out to me, my email's there on the screen, or you should have you got an email from me to get into this. So shoot me an email. I'd be happy to follow up with you and get you some more information, work with you on how that might be helpful to your system. Shiva says, is your study done based on foliar applications or soil applications for avocados? Both have been done in avocados. However, the majority of fertility inputs in avocados go in through the soil due to avocados having a very waxy cuticle on their leaves for the majority of the summer months. So the majority of fertility inputs and biostimulants in those cases are soil applied. We use DiCap, Redox, and Oxycom to some extent in avocado production. And we'll use mainstay SI in bloom sprays or thrip sprays when we have more susceptible tissue to foliar nutrition on the plant. So yep, that's that one. Derek, can DiCap replace KTS or other K sources? If so, at what rate? I heard 3 to 5 pounds a month for 3 to 4 months, or used in conjunction with that KTS. If so, how does that rate change per 10 gallons per acre standard? Looks like we need to start earlier this year. Yeah, so DiCap, we've used it in conjunction with KTS as well as in replacement of KTS in a lot of different crops. In avocados specifically, which I think is what Derek's referring to, and probably lemons here as well. We've done a lot of work looking at, ran for a couple of years where we maybe cut our KTS rate in half and take that and put that into 3 or 4 pounds of, in that case, probably 1 to 2.5, 3 pounds of DiCap, and have been very happy with that. One of the nice benefits we get there is we still maintain the potassium input. However, we get that added antioxidant production from that. One thing that we've done the last couple of years here is we've dropped KTS altogether out of our programs and switched to a heavier DiCap application, that 3 to 5 pounds that I mentioned earlier, and that has served us very well. It has the benefit of reducing our overall EC load that we're introducing to the soil over the summer, which is very important, especially with our decreasing water quality these days. But maintains that efficacy. The other nice thing is, and we don't talk about this enough, I think, is we're also bringing nice amount of phosphorus in, which we don't usually do over the summer months, but can still be very beneficial to our crop. So we use that quite a bit. Yeah, so both strategies have been used, both have been successful. We did a lot last year at Pine Tree Ranch where we did just high DiCap rates all summer, that kind of 3 to 5 pounds, kind of peaking out when it's the hottest in July through August. Kind of going with that 5 pound rate, which is also our peak potassium need, is over those summer months. So that's a nice correlation there. And then tailing off in the late summer to maybe that 3 pound rate has been our strategy. Natalia asks, do you have information or results on the yield of fruit trees? We do. We've done a lot of work with these products in assorted different fruit trees, from cherries to apples, and avocados and citrus and so on and so forth. So if there's a certain fruiting crop you're interested in, then we will, then that's just shoot me an email and I can help you with that. Bob asked, should we supply our CCA numbers through the chat function in order to receive our 2 CCA hours? Please don't. I will send you the CCA sign-in sheet so you can submit it through your own through your own system, through the barcode, and that way you can report it through that. So this has been approved for 2 hours of CCA units. So since you were here, we will send that through in our follow-up email. Everybody who attended will get a copy of the sign-in sheet that they can then sign in. So very good question, Bob. Thank you. That was very important. I know a lot of you were here for those 2 hours, and I really appreciate your guys' time. So thank you. Okay. Let's see, I've got some questions over here in the chat. Let's see, plan. Sorry, I got to figure out where we started here so I kind of don't miss anybody. Okay, here we go. So, can you explain how microencapsulation improves? Oh, I already got that one. Okay, most people do not believe in SI as foliar efficient uptake. So that's a great point. So a lot of people don't, and that goes for calcium as well. And a lot of that goes down to that reactivity of that of those products when they're in their ionic form. Calcium 2+ and silicon silicic acid are both very reactive, and when they're sprayed foliarly, a lot of the times they react and they're not plant available at that point. However, with the microencapsulation technology that we have in Mainstay SI, it maintains that availability, and the plants can take them up and utilize them translaminarly. And we do a lot of foliar applications of Mainstay SI. There's thousands of acres that get treated with Mainstay SI every week in California as a foliar application with great results. So it is unique. We have some trial work comparing different calcium inputs where Mainstay SI was improved, and Mainstay SI was one of the few that are the only one that actually consistently performed to increase the metrics that we'd like to see on foliar applied calcium and silicon uptake. So we have high confidence, I guess, and sell a lot of Mainstay SI into foliar applications. Jim, if romaine plants are susceptible to INSV, would Oxycon be better prevention than calcium silicate? So that's an interesting question, because there's kind of two things going on there. So INSV is impatient something virus, necrotic spot virus. There we go. And it's a huge issue where it stunts lettuce plants, and it's been a nightmare. The last year was horrendous for this in the Salinas Valley, and this year is not looking any better. It's actually looking to be worse this year. So so Mainstay SI, we actually have some research from onions and other crops where if Mainstay SI is used in there, we actually see slightly less thrips damage on the leaves, and that's supported through other silicon products as well, where when there's silicon there, we just see a little bit less thrips feeding damage. However, is that enough to reduce transmission? And that I'm not sure. So less feeding by thrips is beneficial. However, is it enough? And that I'm not sure. And we have not done any specific work at INSV. Now, we have seen with other viral issues, say in potatoes or sugar beets, where Oxycom is used, we've seen less incidence of those viruses showing up within the fields. But a lot of those viruses are more so dependent on having crop stress present for them to be symptomatic, whereas INSV seems to be a pretty capable virus at causing problems whether there's crop stress or not. So we're doing some demos right now with Oxycom early season on kind of that first cover spray within lettuce to see if that will help the plants grow through the INSV stress. So we'll see where that goes. So Jim, I'll touch base with you and we can kind of talk about that, because I'd like to try and get that rolling out in a few more places and see how it goes. Okay. Tracy left. Thank you, Tracy. I'd like to have, this is from Eduardo, I'd like to have more details about how to use Mainstay SI for producing. Oh, you already, that's sent over. John, bolstering my cannabis comment, saying I sell a lot of Mainstay SI into cannabis. Yeah, so that's the questions I have in the chat. I had a couple others that I thought I'd throw up here for you guys. So that usually people send in when I give this talk, and when exactly should I put this on is one that I get a lot. And the timings are going to vary depending on the crop, the goal, the stress, the product that's of interest, right? So for example, Mainstay SI, is something that needs to be there while you're building the tissue for that crop. So for example, in berries, that really needs to be there the first week or two after pollination. That goes for any fruiting crop if you want to have an impact on the fruit quality. So we apply it in strawberries every week because there's always fruit that are in that stage throughout the fruiting process. So we'll do a pint every week. Something more like an avocado, which you see in the picture here behind the slide, they have a little bit more succinct of a fruit development cycle, or something like almonds where it's a lot more tight, or even table grapes or something like that, where you have these really tight crop production cycles. We'll do mainstay SI at pollination, so in a bloom spray, as well as 1 to 2 weeks after pollination, and that has been very effective. We've done that in wine grapes a lot, where we've come in right after when we start seeing petal fall in wine grapes, and come in with a quart of mainstay SI, and that has been a great tool to help increase just that skin integrity all the way through harvest, where we see with that single application at bloom, we'll see less sour rot, things like that that are related to cracking and berry quality issues all the way down at the end of summer when we come into harvest. Redox is one of those where if it's just I want to help my plant deal with stress, maybe you put a little bit out every week or two or month or something like that. If it's I have a huge root stress issue, it's let's go with a higher rate, like a half gallon, to try and help push better root growth right now. Same thing's true with oxycom or dicap. It's kind of dependent. How do I get my CC errors? Bob already answered that one. Dicap looks like expensive MKP to me. That's one I get actually a lot in the field. There's a big difference there between MKP and dicap, I guess, right? So they both have K and P in them. However, when we make dicap, we utilize a lot of amino acid technologies, as well as, like I said, some of the oxycom technology to build a product that has the complex chelation of a high efficiency nutrient product, as well as helping the plant deal with or pushing antioxidant production. So MKP is a straightforward monopotassium phosphate, and that phosphate binds up in the soil with calcium very quickly. The potassium is present, but will also be buffered out of availability within the soil fairly quickly, or not fairly quickly, but eventually. So we so we have lots of things there that are kind of separating between dicap and MKP. Thanks, Chris and Tanner and Gordon, for hanging on here. I don't see a whole lot of questions coming in for you guys, but I really appreciate your guys' time. Yeah, thanks. I thought it was good. So other questions? Sounds expensive. We didn't talk a whole lot about pricing here, but most of these products are priced to fit within the standard kind of fertility input, the application rates and things like that fit within our standard fertility practices, depending on kind of what our goal is. A lot of the concern around price is really about what's my return on investment, is I think how I really want to frame that discussion. And the most expensive fertilizer is one that doesn't work, right? So that's why I think there's a lot of hesitancy around biostimulants in general, is because some of them just don't work. Especially 30 years ago, there was a lot of things that people called biostimulants on the market that were literally the definition of snake oil. So there's a lot of hesitancy to use some of these products. But we've documented and are very comfortable with a lot of these application rates that we have, that they will have that return on investment further down when we come to harvest. So that's the value proposition is there on these products to help build a stronger plant that will allow you to build a stronger yield. I really like what you're saying, but I already use seaweed and phosphites in my program, which I think do the same thing. Why should I use these? So those, I would say, were the two classic biostimulants that were utilized in the market, and they're also the jack of all trades, I would say, and maybe overused in some situations. They're good inputs. They have their time and place. However, we've found that applying these products with sound agronomy can outpace a lot of the generic seaweed and phosphite input recommendations. Where can I purchase these products? We have an assortment of distributors all over the US. And that has, so if you need help finding a retailer for it, let me know, and let me know who you work with, and we'll figure out who you can get it from, or ask your ag chem retailer, and they'll hopefully be able to find it or direct you to where you get it. I have a question here from can this replace all my granular fertilizer? So it depends on what you're growing, I guess, and what your infrastructure system looks like. But we've done some work, for example, on head lettuce down in Yuma, looking at where most of their fertility is done with granular fertilizer that's spread pre-plant and then applied. And we've done a lot of work where we've put out products at the same timing with sprayed down products instead of spread products, and have found that they have performed just as well as a lot of the granular fertilizer. So in most cases, yes. I'm sure there's some cases where that's not true, but in many cases we find that these products and others within our portfolio are very good at replacing granular fertilizer and being a little more strategic with when and how we put things in. We've utilized all 4 of those products in systems where we've, in say like strawberries, where we've completely removed the granular pre-plant fertilizer and been able to maintain yields and have control on our fertility inputs throughout the system, all the way through harvest, or all the way through the season, without having granular in there. So happy to discuss that more with you. Let me know. I'll follow up with you in person. We can talk about your specific system. So I like this one. This sounds too good to be true. How can these products help with so many facets of production, right? So, and I think that's a sound discussion, because there's a lot of products out there that promise the world, right? Like, so it's and they have a and I guess they promise the world because they can't figure out which facet of production they actually can impact. We've done a lot of work on these to really hone in on where they're useful and how they work. And example like Mainstay SI, it does impact a lot of facets of production. That's because literally when you have a cell wall and a cellular structure that is more robust, you end up with a stronger plant that deals with everything slightly better. So it's based in science and shown in the field. So it's one of those things where if you have that problem, if you have that issue, we're happy to talk to you and see where it might fit. Which one of these can I spray, and can I mix them with pesticides? So all of these except Redox can be sprayed foliarly on the plant. They can all be mixed with pesticides of your choosing. And I don't think we have any noted tank mix restrictions in fungicides or miticides or insecticides or herbicides. So we do a lot of Redox and herbicide applications pre-plant, for example. We do a lot of Mainstay SI and dicap with fungicide applications. We do a lot of Mainstay SI and dicap with miticide applications. So a lot of these are rarely going out by themselves as a nutritional spray. They're generally going out with some type of IPM product as well. I like the concept of silicone, but I tried some and it took me 3 hours to clean out my tractor when it clogged my heads. Well, that happened again. So Mainstay SI doesn't do that. We kind of talked about that earlier with one of the other questions, was that because of the microencapsulation technologies, we don't end up being reactive in the tank. So that has been a noted benefit, that unlike other products, it doesn't have reactivity problems. Now, one thing that is true of all products, I mean, this is a general general spraying guideline, is you should always rinse out your tractor when you're done and clean your lines and everything like that. So here's another question. Can these products be applied at the same time? Yes, many of these products can be tank mixed. It kind of depends on your goal. For example, we will put out Redox and dicap together over the summer months to help build roots and provide P and K and other attributes to the plant. I don't like mixing things in my fungicides. Do you have experience tank mixing these products? So like I just said, kind of piggybacking off the last question, I would say of the foliar applied dicap, which is probably 80% of the dicap that's sold, majority of that is being piggybacked on some type of fungicide application. So to get their P and K out there. So no problem, I guess, piggybacking. Obviously, tank pHs are important. Check all that, all your standard, agitation and things like that, and mixing orders. All of that is important to maintain viability. But chemically, there's no issue with dicap tying up your fungicide or something like that. In fact, it's quite the opposite. Dicap will help that plant, which will in turn help your fungicide be more efficient. I tried Oxycom years ago and loved it, but then it was 2 liquids, and what has changed? So several years ago, we did a formulation update on Oxycom where we took the same technologies and moved them out of liquid formulations into a dry that was a lot safer to handle, as well as a lot easier to transport and cheaper to ship around as a dry. So we're not shipping around carriers, waters. What's really interesting is, though, if you get the Oxycon product, you look at it, it actually looks like salt and pepper mixture. There's several different dry products that are non-reactive in their dry state, and then you add them to water, and there's actually some active chemistry that goes on that produces the actual active chemistry that will influence the plant's antioxidant production capacities. So that's what has changed there. So same product, just a different package. Can you come take a look at my orchard and give custom recommendations? I've got that a couple times. Yes, that's my job. So I'm happy to work with you guys on your ranch to make sure that when you guys get the products, you use them to the best way that will benefit your operation. If I want to try one in my berries, which one would that be? I noticed there's a lot of berry growers on this. And my number one go-to in berries is always Mainstay SI. It has a consistent improvement in crop quality, and crop quality is always a concern in berry production. So dicap is also a great PK input. Redox is great for building a stronger root system. Oxycom is great for helping the plant deal with stress and pushing size and reproductive growth and all these things. But Mainstay SI is usually my first stop for berry production. Do I need all 4? Depends on what your level of stress is in your crop, what your goals are, and what you're trying to achieve. But rarely do we use all 4 on one ranch, but they all do have their place, and they can all be utilized together in a comprehensive strategy. We use a lot of 3 of those products, so we might leave one out, or vice versa. So yeah. What is the packaging size? That's a good standard question. Mainstay SI, Redox all come in 2 by 2.5s, as well as totes if you get to that scale. Dicaps in a 20-pound bag, and Oxycom is a 40-pound bucket. Heat stress in summer, what's the rate and how far ahead should I apply? I think we already had this question once, and so it's really 3 to 5 days ahead of time. 2 to 4 pounds, usually 3-pound rate is where we're at for summer heat stress on vegetables and things like that. And that's usually as a foliar application, maybe on that 4 to 5 day range, water run, maybe 4-pound rate, something like that would be about where we'd be at dicap. A follow-up on this that I've had is, if I'm already using dicap and it's going to be really hot, do I need to put more dicap out? And the answer is, it really depends how far away your dicap application was from when the heat event is, and generally we just go with a slightly less rate of dicap when we're having right before that heat impact. Yeah. Will this be available to view later? This was all recorded, so Sam's gonna get this all cut up and figured out, and then it'll get on our YouTube page, and we'll send it out to you in the follow-up email. And how do I get in touch? My phone number and email is right there. Text me, call me, email me, whatever works best for you. I'm responsive, and I look forward to working with you guys this coming year if some of these things sound exciting to you. So that's what I had for my frequently asked questions. If you had any more, feel free to shoot them over here in the chat, or give me a ring, shoot me a text, send me an email, whatever works for you. And I'll be following up to see if you're interested in working with any of these products within your production systems. So thank you so much, and we'll talk soon.